we have two exciting talks today that have one thing in common. They both talk about reactive subtypes or reactive substates of astrocytes. And so just so we're all on the same page, when we talk about astrocyte reactivity, we're talking about changes at the transcriptomic or proteomic level, or the sort of cell physiological level or morphological level of astrocytes in the context of a pathological insult. And so the two papers today are going to show quite nicely something that we've sort of discovered in the last couple of years, is that the reactivity state of an astrocyte can sort of be extremely heterogeneous. It can fall into a distinct reactivity states, and this depends, and we're going to see this today, partly on the type of the insult. We're going to see a nice paper from Brendan showing a sort of acute inflammatory insults, as well as Philip's were sort of showing like chronic insults to the astrocytes, identifying therapeutic pathways in those astrocytes. And I think we're going to start with Brandon, is that correct?
Sure.
Yes. So Brandon, I'm going to give you a wee introduction. So Brandon did his undergrad, his bachelor's at Yale, and then tagged in Martin Campbell's lab at UCSF, where I think he generated all this lovely data he's going to talk to you about today, but is now a PhD student at Harvard. And Brandon, I'm very much looking forward to your talk. I read your paper, really enjoyed it, and I'm looking forward to see how you tell the story.
Great. Thanks so much, Philip. That was a really lovely introduction. And so just checking that there's no technical difficulties with slides here, you're able to see everything.
Yeah, that's good.
Great. Yeah, well, thanks again for that introduction. And thanks again, Jerry, for inviting me and all the other organizers of the Open Box Science Seminar. You know, it's really special, especially as a young trainee, to be invited to this. And I'm super grateful and really just excited to talk about this story from, as Philip mentioned, when I was a tech in Martin Kampman's lab at UCSF. And we uncovered what I think is really kind of an unexpected role of lysosomes in the pathological functions of inflammatory reactive astrocytes. And so for the non-neuroscientists in the room, or in the virtual room, as it were, you might be thinking, why are we spending so much time on astrocytes today? And I'm sure at least you're familiar with this idea that the brain is host to a truly diverse set of neurons who, through their different connectivities and functional firing properties, give rise to all the interesting computations and higher order functions that we ascribe to the brain. And with all this interesting biology to be discovered at the neuron level, it can be quite easy to forget that neurons really only contribute 50% of the total cells to the brain. And so, for example, I'm highlighting only a few other cell types here that you might find in the brain. For example, the brain is highly vascularized, so you'll find endothelial cells throughout the brain, and you'll find various types of glia, such as oligodendrocytes, microglia, and of course, today we're going to double click on the most numerous non-neuronal cell type in the brain, the astrocyte. And these cells are really cool in that they carry out some processes that are just so central and fundamental to the function of the nervous system and physiology. And I've highlighted just a few of those at the bottom of the screen here, such as recycling neurotransmitters at the tripartite synapse, phagocytosing debris, and responding to and resolving insults that the brain may come across during life. But as you can imagine and maybe predict, this intimate relationship between neurons and astrocytes can be co-opted in disease contexts in which astrocytes adopt this reactive state that Philip introduced so beautifully there. And in particularly, we see these reactive astrocytes in a host of neurodegenerative pathologies, and particularly I would note in those with an inflammatory component. And this reactive astrocyte is characterized not only by a loss of homeostatic functions, but also importantly for what I'm going to talk about today, gain of toxic functions, which include the secretion of overtly neurotoxic substances. And so that kind of motivates the broad question, is that we know these things exist, but what are the molecular and cellular mechanisms that govern the deleterious functions of reactive astrocytes? And in the Kampman lab, how we address this kind of broad question is asking, how can we leverage these large scale, super rich omic data sets to understand very specific intractable cell biological or functional changes in specific cell types, and then ask in turn, how do these functional changes perturb cell interactions with their neighbors and ultimately how might these processes be involved in disease. And so our simplistic model to begin to answer this question, we did in human-induced pluripotent stem cell-derived astrocytes. And we have an immense debt of gratitude to Philip's mentor, Shane Littleow, and many others in the field who in the past few years have demonstrated that 3 microglia-derived cytokines, IL-1 alpha, TNF, and C1q, are necessary and sufficient to induce a reactive astrocyte state, which is reminiscent of what we see in many pathological conditions, for example, various neurodegenerative diseases or aging potentially. And just based on time, I don't have the bandwidth to go into all the details about our model, but I think I would direct you to a preprint that came out right around the same time as this mTOR story I'm going to tell you about today, in which we go through in detail the functional relevance and validity of this model in various detail. But suffice to say, for what I'm going to talk about today, a really key aspect that is maintained in our model, and previous work was largely done in mouse, is the secretion of an overt neurotoxin. And so first, to get a handle on what's going on in astrocytes in our model, the first thing we did was obviously conduct RNA sequencing on reactive and non-reactive astrocytes. And the initial data looking at genes that are upregulated may not be surprising to you, those that are familiar with the field. For example, we identified NF-kappa B pathways upregulated, and of course, as we're treating with cytokines, some of those pathways go through the roof too. But what's really interesting is when you look at what's downregulated. And as you can see here, as I've highlighted with these red arrows, many genes that encode proteins that are localized to lysosomes are strongly downregulated in this model. And this actually was also seen in other published data sets, which I'm not showing here though. And so we wanted to move this one step further and ask, are there functional changes to lysosomes? As we all know, you sometimes can get duped by RNA-seq, right? And there's tons of post-transcriptional mechanisms. But I have to admit that some of the really early studies we did were quite disappointing. As you can see here, markers of lysosomes, LAMP1 and LAMP2, show no difference in total lysosomal mass or distribution or size, or any metric you want to use between reactive and non-reactive astrocytes. But kind of our first hint that lysosomes might actually be different between reactive and non-reactive astrocytes is when we stained with Lysotracker, which is a fluorescent dye which accumulates in acidic organelles such as lysosomes. And this was concomitant with a massive upregulation of LC3, important autophagy protein, which is degraded in the lysosome. And so with this, we had some hints that lysosomes might be different, but we didn't really have a good grasp on how or why or what was contributing to this. And so to kind of probe this in an unbiased way, we did lysosome-specific proteomics on the lysosomes of reactive and non-reactive astrocytes. And I'm sure you can imagine this was an incredibly rich data set, and I'm only going to highlight 2 kinds of trends we saw in the data that really jump-started a good portion of the project. And so the first observation we made is that many proteins that you would ascribe to being super central to the main function of the lysosome, which is degrading substrates, were depleted in the lysosomes of reactive astrocytes. So here I've highlighted a few examples for the cathepsin family, cathepsin D, downregulated in reactive astrocyte lysosome, and importantly, subunits of the vacuolar ATPase, which is responsible for acidifying lysosomes, which again is central to its degradative function, were also downregulated. And so this led to the hypothesis that maybe there's not a difference in the mass of lysosomes, but they're functionally different in their degradative capacity. And so to test this, we collaborated with Amy Gao at UCSF, who quite literally just downstairs from us, who developed this Firefly reporter, which is a ratiometric probe, which specifically assays the pH of lysosomes. And hopefully what you can see from this graph here is that indeed in reactive astrocytes in red compared to vehicle-treated controls, there's a significant alkalinization of lysosomes. Another way to say this is that they're less acidic, obviously. And secondarily, we introduced this widely used autophagy reporter, the LC3-GFP RFP reporter, which reads out autophagic flux and is a readout of lysosomal degradative capacity, which, as I mentioned, is a central function of the lysosomes. And again, we got this really kind of striking result in which, compared to our negative control here, bifidobacterium A, which completely inhibits lysosomal function, you can see that reactive astrocytes are only able really to recover maybe 40, 50% of their lysosomal degradative function. And so this was quite confusing actually for a while. And it's that, coming up in undergrad and stuff, you learn the lysosome degrades things and it's acidic, and then you kind of move on. And so we found that in our model in reactive astrocytes, they're doing neither of these things. So what are lysosomes doing in reactive astrocytes? And that brings me to the second finding that I think was really important from our lysosomal proteomics experiment. And that's that exocytosis machinery seemed to be selectively recruited to the lysosomes of reactive astrocytes. And so just kind of in a very simplistic way, we asked if it's possible for lysosomes to maybe in an adaptive way when they can't properly degrade things, fuse with the plasma membrane and actually get exocytosed as a means to deal with their cargoes. And we certainly weren't the first people to ever think of this or study this. And so we were actually able to leverage tools from other groups that use this cell surface LAMP1 staining as a means to assay lysosome exocytosis. And just to be a little bit more specific about what this is that when a lysosome exocytoses, it will fuse with the plasma membrane. And importantly, the luminal cargoes will be expelled to the extracellular space, but what will remain on the plasma membrane is the luminal domains of these transmembrane proteins, for example, LAMP1. And so by developing antibodies against the luminal domains of these lysosomal proteins via flow cytometry, we can read out levels of cell surface LAMP1. And here, I hope what you can appreciate is that in the middle panel, relative to an IgG isotype control, there's quite substantially upregulated levels of cell surface LAMP1, which is again is indicative of lysosome exocytosis. And just a technical point, these cells are obviously not permeabilized in this paradigm. And then, so to kind of complete the thread here by connecting our observations that lysosomes have disrupted pH balance and that they execute lysosome exocytosis, we formally tested this hypothesis by treating with bifilmycin, which I mentioned induces lysosomal alkalinization. It inhibits acidification. And what we see here is that in agreement with our model, with increasing doses of bifilmycin, we're able to progressively increase the pH of lysosomes, and this is concomitant with a substantial increase in cell surface LAMP1 or lysosome exocytosis. And I should note that this isn't just resting astrocytes, this isn't in reactive astrocytes, demonstrating that lysosomal alkalinization is sufficient to induce lysosome exocytosis. And so just to loop back to our original motivation here, I hope I've shown you just a portion of the data that we include in the paper, in that one functional outcome among surely many of reactive astrocytes is that they execute lysosome exocytosis, presumably in response to lysosomal remodeling or alkalinization or whatever you want to call it. And so something that might be going through your head, and certainly was going through our head at the time, is why should you care about this? And so the next question that we tried to probe is, how is lysosome exocytosis involved in the known deleterious function of reactive astrocytes in our model? And we hypothesized that because, as I mentioned, when lysosomes get exocytosed, they're contributing to the secretome of reactive astrocytes, that this might be involved in some of the non-cell autonomous functions of reactive astrocytes, namely neurotoxicity. And so to formally test this, we we instituted the following paradigm, in which again, here in gray, let me see if I can get a laser pointer up. Hopefully you can see that. In gray, we have our iPSC-derived astrocytes, and then treating with a vehicle control or triple cytokines. Here we induce reactivity, and then we take the conditioned media, but not the cells themselves, and throw that onto iPSC-derived astrocytes. And our readouts are twofold. The first is in astrocytes, in which, again, we stain for cell surface LAMP1 as a proxy for lysosome exocytosis. And the second readout is we take the neurons and we assay cleaved caspase as a read of neurotoxicity. And we also did orthogonal methods of reading out neurotoxicity. And so just as a reminder here, just at baseline without any other perturbations in reactive astrocytes, this is what the data will look like. We'll see an increase in cell surface LAMP1, and recapitulating many studies in the literature, we see over astrocyte-mediated neurotoxicity. And next we asked, what happens if we inhibit lysosome exocytosis? And so we made use of this small molecule, vacuolin, which in the literature is a specific inhibitor of lysosome exocytosis. And I hope what you can appreciate here is in the top panel, despite what I would call a modest decrease in lysosome exocytosis, we're able to decrease the astrocyte-mediated neurotoxicity on the order of maybe 50, 60%. And secondarily, we asked the converse question, which is, what happens if we increase lysosome exocytosis? So again, we made use of bifilomycin as our tool to increase lysosome exocytosis in both vehicle and ITC. ITC is the abbreviation we use for IL-1 alpha, TNF, and C1q, treated astrocytes. And this is, I think maybe one of the most surprising things in the entire paper is when we look at neurotoxicity, what happens is that only reactive astrocytes treated with bifilomycin become more toxic. And vehicle-treated astrocytes in this bar do not become more toxic, even though they have substantially elevated levels of lysosome exocytosis. And I think, admittedly, we still don't understand why this is, but I think it suggests that the actual cargo and content of lysosomes in reactive astrocytes is somehow different between the two conditions, and that's what gives this delta between the bifilomycin and DMSO-treated control. Okay, so again, coming back to why should you care about this? I think we show some other data in the paper, but I think even from a cursory glance, you can see that in some way, lysosome exocytosis appears to be connected to astrocyte-mediated neurotoxicity. And from a mechanistic perspective, what I was interested at this juncture, here I'm just showing again the schematic of how we think things happen in reactive astrocytes, which is alkalinization leads to exocytosis and somehow leads to downstream neurotoxicity. But what I was interested in is molecularly, what's kind of the upstream cascade here? Is there some sort of factor X that reactive astrocytes work through that we can blunt and perhaps ameliorate this entire cascade? And so for the rest of the talk, we'll tell you about how we discovered mTOR by CRISPR screens as a modulator of many elements of this pathway. And so just to quickly breeze through some of the methodological considerations here, we took our iPSC astrocytes and endogenously expressed CRISPR-I machinery and then transduced them with a lentiviral guide RNA library targeting genes involved in the druggable genome, as it were. And then, as before, I've shown you, we treat these astrocytes with either vehicle control or triple cytokines, and we stain with either lysotracker as a proxy for lysosomal pH or cell surface LAMP1 as a proxy for lysosome exocytosis. And then using fluorescence activated cell sorting or FACS, we can specifically isolate high, populations of astrocytes that have high and low levels of these fluorescent markers. And then by collecting these populations and isolating the guide RNAs within these cells, we can begin to get a little bit of an idea about the genes and pathways involved in modulating both lysosome acidification and exocytosis. And so just at 30,000 feet, I think the screen here demonstrates what I showed you a specific example of, which is that lysotracker and cell surface LAMP1 are inversely correlated. So what this shows is for any given gene, if say for a given gene you have low levels of lysotracker indicative of alkalinized lysosome, perturbing that same gene in the screen tended to lead to increased cell surface LAMP1. And then if we dive into this a little bit more about what are the specific genes that have the strongest effects on both cell surface LAMP1 and lysotracker, what we found was that many genes involved in the mTOR signaling pathway had very strong readouts on the screen. And so the first question we want to know is that endogenously, without perturbing genes or anything, what is mTOR doing in reactive astrocytes? And I hope you can appreciate from the Western blot on the left and the quantification of such in the right is that by looking at downstream targets of mTOR and their phosphorylation status, we were able to conclude that there seems to be broad mTOR activity in reactive astrocytes. And I'm sure everyone is aware that mTOR is a highly druggable target, and that originally was at least part of the impetus for the guide RNA library that we included, was that, as I mentioned, it was against genes in the druggable genome, meaning that there are small molecules inhibitors of it. And so to follow up, we asked if we use this pan-mTOR inhibitor, PP242, are we able to blunt this entire pathway and potentially ultimately rescue neurotoxicity? And so in the next few slides, I'll just walk through that data. And so the first thing we asked is what happens to the pH status of lysosomes. And so again, here we're using the Firefly ratiometric reporter from Amy Gao's lab. And what you can see is that in both vehicle and reactive astrocytes, there's a dose-dependent decrease in the pH. That is to say, lysosomes get more acidic with more of this inhibitor. And this is true, again, in both vehicle and reactive astrocytes. And next, we looked at lysosome exocytosis. And what's really amazing here, again, we're looking at our cell surface LAMP1, is that at similar dose ranges where we begin to acidify lysosomes around 100 nanomolar, we're able to essentially completely suppress lysosome exocytosis, much more than actually published compounds such as vacuolin, which were lysosome exocytosis inhibitors. And lastly, we tracked these same concentrations of drug, and as I mentioned earlier, use the paradigm where we take the conditioned media from astrocytes and ask what does that do to neurons. So here the y-axis is a readout of neurotoxicity. We're looking at cleaved caspase in reactive astrocytes. And I hope what you can appreciate here is that in the dotted line with the drug, but not the DMSO control, at the highest concentrations, we essentially completely rescue this astrocyte-mediated toxicity. And again, just maybe a technical note, but I think an important one here is that for this data and any other small molecule data that I've shown in this talk, we also have conditions which we call unconditioned media, in which we add the drug directly to the neurons without ever seeing astrocytes. And none of these compounds have any effect on neurons directly, demonstrating that this delta that you see here between the DMSO and PP242 condition can be attributed to the role of astrocytes. And just lastly, to close the loop here with a genetic perturbation, we knock down mTOR itself via lentiviral methods. And in lysosome exocytosis, you can see that specific to the ITC condition, we essentially completely rescue the increase in cell surface LAMP1 in reactive astrocytes, and concomitant with is essentially a complete rescue of the astrocyte-mediated neurotoxicity, again, as read out by cleaved caspase in iPSC-derived neurons. And so quickly, I hope I'm doing okay on time here. I'll just give you a quick summary of things that hopefully I haven't rushed through too much. And the first thing that I think is really new and exciting is that lysosomes are involved in reactive astrocytes. And the lysosome remodeling is characterized by alkalinization, and secondarily lysosome remodeling and alkalinization leads to increased lysosome exocytosis in reactive astrocytes. And as I mentioned previously, this isn't somehow connected to neurotoxicity. And I think I want to make a specific point here about what we're not claiming. And that is that we're not claiming that what's in the lysosomes is the direct toxic molecule. You can see in the schematic, it's completely plausible with our data that things that get exocytosed in lysosomes work in an autocrine paracrine fashion on lysosomes, and the toxicity is through a completely orthogonal route. And I think work from Kevin Gudenplan and others have really moved the needle forward in this, and that's something we're looking into as well. And lastly, the last few minutes, I demonstrated the CRISPR-I screen and how we found that mTOR signaling is potentially a druggable target in which we can control this lysosome remodeling exocytosis neurotoxicity axis. And I think just looking forward, right, I hope what has come across a little bit is the power of the iPSC model is that we can do experiments at scale in a way that you with current technological constraints isn't possible in vivo, and allowed us to do some interesting biochemistry and CRISPR screens, for example. But obviously, ultimately, as with any model, there's drawbacks, and we really value the in vivo work. So right now, we're trying to translate some of these findings into mouse models, both in the Canon lab and in collaboration with others. And secondarily, I think what's a really interesting current line of investigation in the lab is what exactly is mTOR doing? We know that mTOR has quite broad effects in the cell, obviously. And so unfortunately, I had to leave the lab before I could, really crack the code here. But that's something that I think is really interesting. And so lastly, just in a minute, I want to highlight all the wonderful people that I had the privilege of working with on this project. And I think first goes without saying, that includes Martin, my PI, and Kuhn, the MD PhD student that I worked with, and co-first author on this paper. And, as Philip mentioned, I completed this work as a technician, and it's really not lost on me, the amount and emphasis on mentorship and truthfully patience that both Martin and Kuhn probably had to have to give me the latitude and independence to take on this project. And I'm just really grateful for them. And I want to highlight them as amazing scientists and mentors, but more importantly, people. And want to highlight 2 rotation students that worked with me. Indigo Rose did some of the CRISPR screens, and Saeed did some of the IF that I've shown today. And thank you very much to our collaborators as well. I talked about Amy Gao's contributions to specifically the lysosome pH work, and also worked through CZI with Manu Minetti and Josh Elias's lab, specifically with the lyso-IP and mass spec work. And don't hesitate to reach out, and really looking forward to the discussion and questions. Thanks so much.
What a beautiful talk. What a great story, Brendan. If you have questions for Brendan, put them in the chat. Oh, they're already coming in, I see. I think the other speaker, Zoeb, has a question for you. I'm going to just read them out for you if that works for you. So Zoeb asked, do you think this story mechanistically links with the neurotoxic saturated lipids from the Little lab?
So sorry, you cut out a little bit on my end, but I believe the question is addressing some of the beautiful work from the Little lab showing that polyunsaturated fatty acids are the toxic molecule.
Yeah, absolutely. Yeah.
Right? Yeah. So obviously that's intrinsically connected to this, and just such a beautiful paper. And I think one thing I can say is we just got lipidomics data back in our model, and it seems broadly consistent with theirs. And I think it's really exciting actually that the models are converging like this. And one thing that we're looking at in the future is doing this lyso-IP paradigm and seeing maybe if this toxic molecule is actually somehow localized to lysosomes. And these would be like really cool complementary approaches.
Yeah, excellent. We have so many questions. My God, I'm gonna have to pick some. I'm gonna pick Priyas from the Little lab. Priya is asking, can you speculate on how the lysosome remodeling may affect the phagocytic capacity of reactive astrocytes? Is it known if reactive astrocytes have impaired phagocytosis?
Yeah, totally. And yeah, I think that's probably 2 sides of the same coin, right? And so again, I would direct you to Kuhn's first authored paper, in which definitely in agreement with some of the mouse models, there's deficits in phagocytosis in these astrocytes. So certainly pleiotropic roles of lysosomes here, and distilling kind of causality, I think is a really exciting avenue.
Let's do one last question before we move on to Zoeb. Bruno Benitez is asking, another way to measure the lysosomal exocytosis is by quantifying levels or activity of lysosomal enzymes in the conditioned media. Did you perform proteomic analysis in the conditioned media or Western blots for some potential effectors of the neurotoxicity, for example, the cathepsins B and D?
Yeah, totally. Awesome question. And yeah, we were trying to be very sure that if we're making this claim about exocytosis, that we can prove it. And so in the paper, we did turf microscopy, live turf microscopy, to visualize lysosomes and their fusion with the plasma membrane. And we didn't do proteomics, but we did ELISAs for, I believe, cathepsin B and D in the supernatant of reactive astrocytes. And it showed the same trend that I showed here with the lysosome exocytosis, or with the cell surface lamp one, excuse me. And we just went ahead with the cell surface lamp one just because the throughput is a little bit higher and allowed us to do more experiments.
Okay, cool. There's more questions. I'm sure that Brendan would be happy if you send those questions in an email to him. I'm sure he'd love to answer them. But